Hello, I am Rebecca Millman, Associate Professor and Section Chief of Radiological Sciences at the University of Colorado School of Medicine. This presentation provides a brief overview of radiation-induced cellular and tissue effects. In this presentation, I will discuss how radiation deposits DOS in cells, radiation-induced damage and repair, and radiosensitivity. I will briefly discuss radiation effects on tissues and organs. A more in-depth discussion is part of a separate presentation. In X-ray imaging, X-rays are produced within the X-ray tube and directed towards the patient. The X-rays then pass through the patient and interact with the image detector. The radiograph that you look at represents the X-rays that fully passed through the patient and made it all the way to the detector. There are three basic things that can happen to an X-ray photon when it reaches a patient. The first is that the X-rays pass through the patient without interacting with the tissue at all. In this scenario, the photon exits the patient unchanged. In other words, no energy is transferred from the photon to the patient. The second thing that can happen is that the X-ray passes through the patient and in the process transfers some of its energy to the patient. The energy that's not deposited exits the patient as a scattered photon. The third possibility is that the X-ray deposits all of its energy in the patient, and there's nothing left to exit the patient and interact with the detector. When discussing patient dose, it's these last two scenarios we need to consider, because, as a reminder, dose is the amount of energy deposited in tissue per unit mass. It has SI units of joules per kilogram, or Gray. On an atomic scale, there are a variety of interactions that can take place when X-rays interact with tissue. This plot shows the mass attenuation coefficient as a function of energy for soft tissue. The mass attenuation coefficient represents how likely it is that a particular type of interaction will occur between an X-ray photon and soft tissue. I'm not going to talk about most of these, but it's worth noting that the most common interaction that takes place during medical imaging exams is Compton scatter. So let's look briefly at what happens during Compton scatter, since this is the mechanism by which energy is deposited in tissue. Compton scatter occurs when an incident photon, or X-ray, interacts with an orbital electron in an atom. So let's consider what happens to the energy of this incident photon. Some of that energy is needed to eject the electron from the atom. Some is transferred to that ejected electron, also called a Compton electron. This electron goes on to interact with other atoms. The rest of the energy is in the form of a scattered photon, which may go on to interact with other atoms. So in the process of Compton scatter, energy is transferred from the incident X-ray to the ejected electron and to the scattered photon. Between 15 keV and 150 keV, which is the energy range for all X-ray based imaging, including CT, radiography, mammography, and fluoroscopy, most of an incident X-ray's energy ends up as the scattered photon. Because an electron is moved from the atom, Compton scatter is an ionizing event. So this ejected photon may go on to interact with other things, or it may pass through the rest of the patient's tissue without interacting with anything else. The ejected electron will continue to interact with other atoms, creating more scattered photons and more scattered electrons. With each interaction, the new scattered electron has lower and lower energy. This process continues until the scattered electron's energy is so low that it can no longer cause ionization events, which occurs around 10 eV or 0.01 keV. We can model the paths of these electron interactions within a cell. So the incident photon first interacts with the cell through Compton scatter and ejects an electron from the atom that it interacts with. The ionization event is represented here by a dot, red dot. The ejected electron then goes to interact with other parts of the cell, with the electron path represented here by a red line, and each ionization event indicated by a red dot. Energy is not deposited uniformly along the electron path. Rather, it's deposited in clumps at different locations along the electron path. What this looks like and what the biological effect is depends on the type of radiation and the energy of that radiation. Here, we've discussed an electron path that was started when an X-ray photon interacted with the cell to produce an energetic electron. Charged particles such as alpha and beta particles, however, directly ionize tissue and start an electron path in motion. There are a couple of different terms that are useful in describing the biological effect from different types of radiation. The first is the linear energy transfer, or LET. This describes how much energy is transferred to the cell per unit path length. In other words, how much energy is deposited over 1 micron of the electron path. High LET radiation includes protons and alpha particles. Radium-223, which is used to treat bone metastases, is an alpha emitter. The LET is about 80 keV per micron. Low LET radiation includes X-rays and gamma rays and beta particles. This low LET radiation deposits much less energy per unit length, about 0.2 to 2 keV per micron. The other important property is the range of the radiation in tissue. In other words, what is the size of the electron path? Over what length is damage occurring? High LET radiation has a short range, about 50 microns. In other words, all of the energy from an alpha particle is deposited over about 50 microns within this tissue. In comparison, low LET radiation has a much larger range, about 500 microns or half a millimeter. Beta particles can have an even larger range of up to 12 millimeters. The LET and range, which describe how energy is deposited in tissue, help us determine what biological damage will occur from different types of radiation. We'll discuss this more in just a moment. What the damage is from these interactions depends on where in the cell the interaction takes place. Outside the nucleus, ionizations don't really damage the cell because they're backup systems for critical cellular functions that take place here. For example, there are redundant systems for protein production, so if one is damaged by ionization, the cell's overall function is generally unaffected. Where we start to see more damage, however, is when these ionization events take place in the nucleus, and specifically near DNA. When we talk about radiation biology and medical imaging, we're usually concerned with this subset of events, ionization events that damage DNA. So the biological damage caused by radiation occurs through the interactions we've been talking about, ionizations that occur along the electron path through a cell. If these electrons interact with DNA, they can ionize molecules within the DNA. This can occur as damage to a base, and it can also occur as damage to the hydrogen bond between the two bases that make up a base pair. Ionization along the DNA strand can cause a strand break. These can typically be repaired, though, through normal cell repair processes. However, if the break isn't repaired, the effect of the damage depends on where along the DNA strand the break occurs. If it's in a portion that controls production of a common protein, it's not a big deal because there are often other locations that make the same protein. If, on the other hand, there's unrepaired damage to a portion of the DNA that controls cell division, this can be more problematic. Sometimes a single electron will cause two ionization events in the same strand of DNA, each damaging a strand. This double strand break is more difficult for the cell to adequately repair and can lead to carcinogenesis by affecting tumor suppressor genes or oncogenes. At diagnostic energies, double strand breaks are much less common than single strand breaks. The most problematic type of DNA damage is what's referred to as complex damage, and this really is just a combination of all the different types of damage that we discussed. From about 1,000 milligray of X-rays, all delivered at once, there are about 40 double-strand breaks, 1,000 single-strand breaks, and 3,000 damaged bases. So this kind of gives you an idea of what the likelihood is of these different types of DNA damage. Damage from high LET radiation, however, is more likely to be complex and hence harder to repair. Remember that high LET radiation has a much shorter range than does low LET radiation. This means that it's undergoing more interactions per unit area, which increases the probability that a single electron track will contain multiple ionizations within a single strand of DNA. Conversely, X-rays, especially the energies used in diagnostic imaging, are much more unlikely to cause double-strand breaks, but it still happens. It's important to note, however, that only a fraction of the radiation energy deposited in tissue results in chemical changes. The vast majority of the energy is deposited as heat, which is of little biological consequence when compared with the heat generated by normal metabolic processes. Aside from the direct ionization effects we've been discussing, radiation can cause damage indirectly. With low LET radiation, this is where most of the biological damage comes from. About 2/3 of the damage from low LET radiation is from indirect effects. The most common indirect effect is free radical production. This occurs when a photon ionizes a molecule, typically water. Since cells are about 80% water, this is a common interaction. This ionized molecule, H2O+, has an unpaired orbital electron, making it very reactive. That H2O+ molecule fills in its missing electron by combining with the hydrogen atom of a nearby water molecule. With the addition of another hydrogen atom, the H2O+ ion becomes H3O+, and the other water molecule becomes a free radical with an extra orbital electron. Again, this unpaired orbital electron makes the free radical molecule very reactive. The free radical is very likely to interact with any nearby molecules. If this happens to be a strand of DNA, then the reaction can cause some of the DNA damage we discussed previously. Base damage, damage to the hydrogen bond between base pairs, or a break in the phosphate strand altogether. Because free radicals are so reactive, they don't travel very far before interacting with another molecule. Consequently, free radicals only interact with, and therefore damage, DNA if they're produced within about 4 nanometers of the DNA. DNA that's farther away is out of reach of the free radical's harmful effects. It's important to recognize that there are other causes of DNA damage that we experience all the time. For example, mitosis can result in DNA damage, and cellular respiration causes some oxidative damage. In fact, our cells in our bodies each experience thousands of DNA lesions every day. Much of this damage is repaired through regular cellular repair mechanisms. Cells can also prevent the spread or replication of any damage by stopping the cell cycle, or even by initiating apoptosis, both of which prevent any mutations from being replicated. So let's review what happens when ionizing radiation interacts with a cell. We can either end up with heat deposition, which, as I mentioned earlier, is of little biological consequence, or we can end up with ionization events. If these ionization events take place with non-DNA cellular components, again, they're typically of little biological consequence. If they interact with DNA, though, they can cause more substantial problems. Still, sometimes this results in minor damage, which can be successfully repaired. However, other times, we end up with a double-strand break or complex damage. This can result in mitotic arrest or apoptosis, both of which can have biological effects, which we'll discuss in a little bit. The other thing that can happen is that repair processes kick in. Sometimes the repair is successful, but sometimes mutations persist and the cell still has mitotic capacity. It's these situations where we start to be concerned that some of these cells might develop into cancer. And so it's important to recognize that this is a possible consequence of ionizing radiation exposure to cells, but it's not the only potential consequence. So we've seen that these double-strand breaks are the root of much of the unrepaired cellular damage. So it's useful to know how often these double-strand breaks are occurring after exposure to different types and doses of radiation. One method of doing this is by imaging proteins that are needed to repair DNA double-strand breaks. One of these proteins is gamma H2AX. By imaging gamma H2AX, we can observe double-strand breaks and their repair process. These images show the immunofluorescence of tagged gamma H2AX proteins, showing the timeline of double-strand break induction and repair in primary human fibroblasts exposed to 200 milligrave radiation. Groups of double-strand breaks and repair proteins show up after 3 minutes, but are virtually absent by 24 hours after exposure. These data are from a cellular study performed by looking at the number of gamma H2AX foci within breast epithelial cells exposed to 20 milligray of radiation during a CT phantom scan. It shows the number of foci in the control, or unexposed cells, and the number of foci 30 minutes, 24 hours, and 48 hours after exposure. This shows a slight persistence of DNA double-strand breaks, although this was not a statistically significant effect. So this leaves us with a couple of unanswered questions. The first is, at what dose and for how long after exposure are the levels of this protein, that is, part of the repair process for double-strand breaks, occur? The second question is, what does it mean if this protein is still elevated 48 hours after exposure to radiation? Does this mean that double-strand breaks still exist and that cells are still trying to repair them? Or does it mean that the body retains this protein for some period of time in preparation for another irradiation event? These are some of the things we still don't know. Okay, so there's some things that we still don't know. What are some other things that we do know? There are a handful of factors that we know influence biological effects of radiation. One, which we've discussed a fair amount already, is the spatial distribution of how the energy is deposited. We've talked about this here, and we've seen that energy deposited near DNA is more damaging than when it's deposited in other parts of the cell. Dose, which is the amount of energy deposited per unit mass, will also affect the cellular response. We've discussed how cells have repair mechanisms, but these mechanisms become overwhelmed at high doses and are not able to repair cellular damage effectively. What's still unknown, however, is the exact relationship between dose and cellular response. In other words, we do not know at what dose these cellular repair processes become inhibited. This will be part of the basis for our later discussion of radiation risk. Cellular effects also depend on the dose rate. If cells are exposed to radiation over a matter of milliseconds or seconds, the effect is very different than if this same dose is spread out over minutes, hours, or day. This graph shows the fraction of surviving cells as a function of dose, shown here in gray. When dose rates are below around 0.1 gray per hour, or 100 milligray per hour, there's a repair of cellular damage during the irradiation. So we can see that this plays a substantial role in cell survival. The effect of radiation on a cell also depends on the cell cycle stage at the time of irradiation. Progression through the cell cycle is regulated by cell cycle control genes at various checkpoints, indicated here by the red stop signs. For example, the G1 checkpoint ensures that before DNA synthesis, the cell is healthy, including checks for the size of the cell, the availability of necessary nutrients, and the stability of DNA. The P53 protein stops the cell cycle at these checkpoints, if necessary, to allow for DNA repair. P53 plays a role in DNA repair and induction of apoptosis if the cell is not repairable. Deficiencies in P53 function have been associated with increased cancer risk, since the cell control mechanisms it typically regulates no longer work properly. The cell is least sensitive to radiation during the S phase and most sensitive during mitosis. The biological effect of radiation exposure also depends on the radiosensitivity of the particular cell or tissue type. So what do we mean when we use the term radiosensitivity, which I've already used a bunch in this presentation? It's defined as the loss of clonogenic capacity in normoxic conditions. So this is very different from the propensity to cause cancer, which is sometimes used interchangeably when talking about how radiation affects cells. General differences in the radiosensitivity of cells was described by Bourganeau and Thibodeau in 1906. I actually have no idea how their names are pronounced, so I probably completely butchered it, so my apologies to them and their ancestors. Anyway, their studies on animal spermatogonia led them to theorize that cells are more sensitive to radiation if they reproduce quickly, are metabolically active, and are undifferentiated. There are multiple examples that contradict these general rules, however. For example, tumor cells have a high mitotic rate, but are not always more radiosensitive than their normal counterparts. Another example is actually found in rat spermatogonia. Their mitotic rate is the same in juvenile and in adult rats, but spermatogonia in young rats are less radiosensitive than those in adult rats. Other examples include lymphocytes and oocytes, both of which are fairly sensitive to radiation, but are not very metabolically active. So here's a list of some of the most radiosensitive cell types. They include spermatogonia, lymphocytes, oocytes, and hematopoietic stem cells. Some of the less radiosensitive cells include central nervous system cells, muscle, and bone. So we've talked at length about some of the effects on cells from radiation exposure, but what about effects overall on an organ? When an organ is exposed to X-rays, the effect on the organ depends in part on the radiosensitivity of the tissues it's comprised of. If the dose is very high, cells within the organ cannot repair themselves and necrosis occurs. At lower doses, radioresistant tissues undergo repair. In this process, damaged cells are replaced by a different cell type. For example, fibrosis occurs. The organ is permanently changed from its pre-irradiated state. Examples of this include tissues within the central nervous system and muscle. Radiosensitive tissues, on the other hand, undergo a regeneration process. Damaged cells are replaced by the same cell type, allowing the organ to heal and return to its pre-irradiated state. Examples of this include skin, the GI tract, and bone marrow. The effect of radiation on overall organ function depends not only on the radiosensitivity of individual cells within the organ, but also on whether the processes within the organ take place in parallel or in series. One example of a parallel organ is the kidney. The smallest functional subunits are nephrons, which function independently of one another. Therefore, damage to one nephron does not affect the function of the other nephrons. And while the overall kidney function may be reduced, it's not completely inhibited. An example of a serial organ is the spinal cord, which I've illustrated here as a neuron. Here, function of a distal part of the spinal cord does depend on the function of other sections. So damage to one section of the spinal cord will result in the loss of function to segments distal to it. Some organs, such as the lungs, have mixed parallel and serial functions. There's parallel architecture of the level of lung parenchyma, where gas exchange takes place, and serial architecture for airways that carry air in and out of the alveoli. So here are some key points from what we've discussed. Ionizing radiation deposits energy in cells. High LET radiation is more biologically damaging than low LET radiation. X-rays are a form of low LET radiation. The most severe damage to cells occurs when radiation causes double-strand breaks in DNA, but most radiation-induced damage is repaired. Radiation causes more damage during mitosis, and some cell types are more sensitive to radiation. While generalizations are often made about cell radiosensitivity, they're not universally true. Radiation effects vary between patients, and we'll see this a little bit more when we discuss radiation effects in a later discussion. And importantly, much is still unknown about the exact mechanisms of radiation dose and cancer induction. Thank you for watching, and if you have any questions or comments, feel free to reach out.